Okay, so welcome back to this next video in which we are discussing ionotropic glutamate receptors. Okay, right, so we've now discussed the three main families of ligand-gated ion channels. And we've discussed that all of the ionotropic glutamate receptors fall into this second family. Okay, so the second family of glutamate-like receptor ligand-gated ion channels, all of these were tetramers. Okay, so they were made up of 4 separate polypeptides stuck together to make the full ligand-gated ion channel. And I now want to redraw out this picture because it's so important, and I'll draw it bigger this time. Okay, and then we'll discuss the different types of ionotropic glutamate receptors. Okay, right, so let's draw the cell membrane here. Okay, then we'll have the amino terminus of the polypeptide of an individual subunit. And remember, I'm drawing an individual subunit. I'm drawing 1/4 of a glutamate receptor. Okay, so here's the amino terminal domain here, which I'll remind you is important in holding the 4 polypeptides together into the whole tetramer. Okay, so here it is in turquoise, like so. Then what we have is the S1 domain, okay, here, and then it'll fold back like so, and then it will have the first membrane spanning alpha helix here. Okay, so this portion that I'm now highlighting in red, this is going to make 1/2 of the clamshell domain, basically. Okay, and this is called the S1 domain. So this is making the upper half of the clamshell, if you like. Okay, and we'll now make the bottom half of the clam shell. Okay, right. Then we have that M2 reentrant loop here, and then we have the M3 membrane region, which is another membrane spanning alpha helix. Okay, and this domain is going to work like so. Okay, so it's going to fold over like so, and then it will come back down like so. So you've got this loop between the M3 domain and the M4 domain here. Okay, and this is known as the M3, M4 loop. So the entire thing, okay, is known as the M3, M4 loop because it's between M3. In fact, I've got so much space I can actually label these things up. This is M3 and this is M4, and I think I might even color code it. So we've got M3 and M4, which I won't do in turquoise because I've already used turquoise for the amino terminal domain. So they're here. And then this entire loop, everything between these two M3 and M4 membrane spanning alpha helices here, is called the M3, M4 loop. I don't want to colour that in because I'm about to colour in the S2 domain. Okay, and I'll just finish off the polypeptide first. You then have the carboxylic acid terminus down here. Okay, so that other important domain then, which made up the base of the clamshell-like domain, if you like. Okay, this is known as the S2 domain here. So this is S2. And now what's going to happen is our ligand is going to bind in here, basically. So our glutamate molecules, or as we'll see, some of the glutamate receptors actually don't. Well, the glutamate receptor subunits, some of them don't have glutamate as their ligand, and we'll discuss that more later. Okay, but here is where the ligand will bind in all cases. Okay, so in purple here, this is the ligand. Right, okay, so let's now discuss the 4 different families of ionotropic glutamate receptors. Okay, right, so the first family then, and again these are not just one receptor, these are families. So we have discussed the ionotropic glutamate receptors now. We've discussed that all ionotropic glutamate receptors are tetramers and they have this basic structure. Okay, that's going to go across all 4 of these families we are now going to study. Okay, so the 4 families then are the Ampa receptors. Okay, that's one family. And the Ampa receptors are often abbreviated to AMPAs for short. Okay, so you put AMPA, and then you have the R at the end, and then you put an S like that. So the AMPAs means the Ampa receptors. Okay, the next family of glutamate receptors that are ionotropic glutamate receptors, and I should stress that there's also G-protein coupled receptor glutamate receptors, which are called the metabotropic glutamate receptors, which we're not going to talk about here. Okay, these are the kainate receptors. Okay, so the second family of ionotropic glutamate receptors is the kainate receptors, and for short, kainate receptors are abbreviated to KARs. Now, Ka doesn't stand for kainate, it actually stands for kainic acid. Okay, kainate is the conjugate base of kainic acid, so don't think that the Ka stands for the two first letters of kainate, it actually stands for kainic acid. It's kind of trivial, but there you go. Okay, right. Then we've also got the Nmda receptors, which are very, very famous. These have gained a huge amount of interest because of their involvement in learning and memory, which we'll see a bit of at the end. Okay, and the Nmda receptors are often abbreviated to the NMDARs, or the NMDARs. Okay, right, so NMDARs. Right, and then there's a final just silly class of receptors, which are put in the category of ionotropic glutamate receptors. However, no one knows what their ligand is. Okay, we have not found a single endogenous ligand yet for this final class of receptors. So you might ask, well, if they're not ligand, if they're not receptors for glutamate, why have they been put in this family of ionotropic glutamate receptors? Well, it's because they have the same structure. They are tetramers, they have this same basic membrane spanning topology and the same sort of arrangement of the amino terminal domain and the ligand binding domain here. Okay, so they're put within this family of ionotropic glutamate receptors, but they're not glutamate receptors, basically. And these are known as the delta glutamate receptors. Okay, and we're going to say very little about the delta glutamate receptors. As I say, it's still not known what the endogenous ligand for the delta glutamate receptors is. It doesn't seem to be glutamate, and it doesn't seem to be anything else that we've tried against these things. Okay, so they're very mysterious. They're an area of ongoing research. Okay, right. So we are going to go through each one of these families in turn. We'll start off with the Ampa receptors. We'll then go on to the kainate receptors. Those two follow each other very nicely because they're almost doing a very similar thing, basically. We'll then do the delta glutamate receptors. We'll put this in here. And then we'll leave till last the Nmda receptors because they're the ones that I've got the most to say about. So we'll leave those till last. Okay, right. So let's start then by discussing the Ampa receptors. Okay, and I won't tell you what Ampa stands for at the moment. I'll tell you right at the end what it stands for. Ampa is basically an artificial agonist for these receptors. Okay, so it's a synthetic agonist, but we'll discuss that later on. Okay, right. So I want to start off by telling you about the different types of subunits for Ampa receptors. Okay, so Ampa receptors is one of these subfamilies of ionotropic glutamate receptors. Now, the simple thing would be if there was just one Ampa receptor. Okay, there was just one subunit for an Ampa receptor called the Ampa receptor subunit, and all you had to do to build an Ampa receptor then was get 4 Ampa receptor subunits, tetramerize them together, and you've then got your Ampa receptor. It's not so simple. There are in fact 4 subunits for Ampa receptors. Okay, so let me now tell you what these are called. So these 4 subunits that are all for Ampa receptors are called GLU-A1. Okay, so the GLU is for glutamate, the A is for Ampa, and then the 1 is just the first member of this family of subunits of Ampa receptors. Then there is also GLU-A2. Okay, and it goes on. There are 4 of them in total. There's then GLU-A3 and GLU-A4. Now, this is the new nomenclature for these 4 subunits which can make Ampa receptors. Okay, these have been studied for a very long time, and therefore there was an old naming system for the subunits of glutamate receptors, and a lot of people are stuck in their ways and still use the old nomenclature. Okay, so the old nomenclature for these 4 subunits of Ampa receptors was to call them GluR1, GluR2, and luckily the numbers match up perfectly. Okay, GluR3 was GluA3. Okay, and then finally GluR4 was GluA4. Sorry, no. GluA4 was GluR4. These are the old names. Okay, so these ones at the bottom, the GluR1, GluR2, GluR3, GluR4, these are the old names for what are now called GluA1, GluA2, GluA3, and GluA4. Okay, so you have these 4 subunits that make up Ampa glutamate receptors. Okay, so all of these different subunits all have the same basic structure here. Okay, they all will differ in their primary sequence of amino acids, so they will all have different sequences of amino acids, but they all fold into something that looks like this, and they are all capable of tetramerizing together to make a full Ampa receptor. Okay, now an important thing to say is that these 4 subunits of glutamate receptors, they can only bind together to make a tetramer with other Ampa receptor subunits. They can't bind together with the subunits of kainate receptors or Nmda receptors or delta glutamate receptors. And that goes for all of these. Okay, there's no mixing between the 4 different families. Okay, even though they have these same very similar structure. Okay, so we now want to discuss how can we actually combine these 4 Ampa receptor subunits together to make a full Ampa receptor. Okay, well the first thing to discuss is the fact that we can build homotetramers. Okay, and tetramer means 4-membered structure. Homo means the same. So this means that the 4 subunits that you are going to use to build your full Ampa receptor, okay, which I'll just draw a picture out just to remind us is here. Okay, so here's the tetramer. Basically, the 4 subunits you are going to use in these 4 slots of your Ampa receptor are going to be the same, basically. So, for instance, if we wanted to build a GluA1 homotetramer, what we would do is we'd make 4 GluA1 subunits, we'd stick them in the 4 slots, we'd then have a GluA1 homotetramer, and that now would be a functional Ampa receptor. It would be a functional ionotropic glutamate receptor. Okay, right. So most of these 4 can actually form homotetramers. Okay, so we know that GluA1, GluA2, and GluA4 are capable of forming homotetramers. So GluA1 can form homotetramers, GluA2 can form homotetramers, and GluA4 can form homotetramers. We have not so far found GluA3 homotetramers. Okay, so these 4 are capable of forming Ampa receptors by homotetramerizing with 3 other identical subunits, basically. Okay, so the homotetramers are the easy ones to understand. There is then also the heterotetramers, which is where you have different subunits within your same Ampa receptor. Okay, so let me draw a picture of this. So once again, I'll draw out this picture of our Ampa receptor, which is a tetramer. Okay, like so. And it will be made up of these 4 separate subunits. So 1, 2, 3, 4. Okay, so we're now going to use different subunits, different Ampa receptor subunits in different slots, basically. Okay, now, so far we have only found what are known as diheterotetramers. And before I tell you about diheterotetramers, I should introduce you to the nomenclature of heterotetramer. Okay, so we've seen homotetramer means that the tetramer is made up of 4 subunits which are all of the same type. Heterotetramer means that the tetramer, the 4-membered structure, is made up of 4 subunits that are not all the same type. Now, diheterotetramers, which are the only types of heterotetramers that have actually been found within nature, are heterotetramers made out of 2 different types of subunits. Okay, so you don't have all 4 subunits being the same subunit, but you don't quite go wild, basically. You only have two different types of subunit within the entire structure. Okay, right. So, the diheterotetramers that we have therefore found are the GluA1, GluA2 diheterotetramer, the GluA3, GluA2 diheterotetramer, and the GluA4, GluA2 diheterotetramer. So basically, if I remind you of the four different Ampa receptor subunits, we had GluA1, GluA2, GluA3, and GluA4. Okay, basically, if you want to build a heterotetramer, you always use GluA2 subunits, and then all you need to do is pick the other type of subunit that you want to include, and you can either pick GluA1, or you can pick GluA3, or you can pick GluA4. So the three different types of diheterotetramers you can form then are the GluA1, GluA2 diheterotetramers, which is a Ampa glutamate receptor, which contains both GluA1 subunits and GluA2 subunits. Now you might say, hang on a second, you're just telling me that it contains some GluA1 subunits and some GluA2 subunits. You surely need to tell me the stoichiometry. You need to tell me it contains one GluA1 subunit and three GluA2 subunits, or two GluA1 subunits and two GluA2 subunits. As far as I'm aware, that knowledge just isn't known at present. Think about how easy, well, how relatively easy it would be experimentally to show that you can find a glutamate receptor which contains both GluA1 subunits and GluA2 subunits. Then think about the complexity of actually trying to compare the stoichiometry. Okay, and even better questions that you can then ask are, if for instance you had two GluA1 subunits and two GluA2 subunits, how are they oriented relative to each other? Is it the case that the two GluA1s are positioned next to each other and therefore the two GluA2s are positioned next to each other? Or will you do it diagonally, i.e. have the two GluA1s diagonally opposite one another and the two GluA2s diagonally opposite each other? Those sort of questions are absolutely not known. Okay. So what we do know is that you can find Ampa glutamate receptors which contain both GluA1 subunits and GluA2 subunits. Okay, and because you've only got two different types of subunit within the whole tetramer, it's called a diheterotetramer. Di for two, hetero for different. So you've got two different subunits within your tetramer. That's where this name comes from. Okay, so you can also form GluA2, GluA3 diheterotetramers. Okay, and this time what you've done is you've paired GluA2 with GluA3. Okay, and then also according to my rules, you can also have GluA2 with GluA4. Okay, so the diheterotetramers we find are generally you take GluA2 and then you compare it with any of the other three. Okay, so we have these three homotetramers then, GluA1 homotetramers, GluA2 homotetramers, and GluA4 homotetramers rather, and we now also have these three diheterotetramers. Okay, those are the Ampa receptors which then are actually known. Okay, right, so now what I want to talk about is what their actual function is. Okay, so what are they going to do when glutamate binds to them? Where are they found? What do they do? Okay, so they are the main receptors for glutamate within the brain. Okay, kainate receptors you find in certain places, but the Ampa receptor is more important than the kainate receptors. Its expression is much more wide. Kainate basically does the same thing, but it's much more limited in where you can actually find it. So basically, if you go to a synapse within the brain, of which there are a huge number of synapses, and this synapse is an excitatory synapse, okay, so there'll be a huge number of excitatory synapses, the chances are that this synapse will be glutamatergic, okay, so that means that the presynaptic neuron, which I'm drawing here, will be releasing glutamate onto the postsynaptic neuron. And the chances are that the postsynaptic neuron will have Ampa receptors on its surface. Okay, so here is our postsynaptic dendritic spine, okay, drawn a little bit oddly, but never mind. So this is our postsynaptic structure. Okay, right, and the glutamate binding to the Ampa receptors that are on the postsynaptic neuron will activate this postsynaptic neuron, basically. It will cause an excitatory postsynaptic potential, and I want to discuss this in a little bit more detail. Okay, so let's draw our Ampa receptor here. Okay, so this in blue, this represents our Ampa receptor. And of course, there will not just be one Ampa receptor on the postsynaptic dendritic spine. There'll be a huge number of them, and they're all going to be being activated by the glutamate that's being released from our postsynaptic neuron. So our postsynaptic neuron is releasing glutamate here. Okay, so this is the neurotransmitter glutamate. Okay, right, so we want to discuss now, when you actually open the Ampa receptor, because glutamate is bound to the Ampa receptor, how is this actually going to depolarize the electrical potential difference across the cell membrane? Okay, right, so for this we're going to have to discuss electrical potential, okay, so that we can understand electrical potential difference. So, let's start off then with electrical potential. Okay, so electrical potential is this concept from physics. It's a mathematical model to help us understand this reality. Okay, and this is the mathematical model. What you do is you ascribe to every point in three-dimensional space a number. Okay, a real number specifically. Okay, this number can be positive, it can be 0, it can be negative, it can be an integer, it can be a rational, it can be an irrational. Okay, it can be any real number. So this is my drawing of a three-dimensional graph. So to every point, so I can pick a random point here, and I can ask, what is the number that you've ascribed to this point? What is the electrical potential of that point? So every point in three-dimensional space has this number associated with it, which is the electrical potential. And in principle, I could have a little man, therefore, okay, shown here, with a machine which can measure electrical potential. And this little man can go over to this spot and get a readout on this machine of the electrical potential at that place. Okay, so that's what electrical potential is. It's this real number that's ascribed to every point in real three-dimensional space, basically. Now, the units of electrical potential are volts. Okay, right. So now let's go from the world of physics to going back to the world of biology. Okay, so let's have a cell here. Basically, all of the points inside the cell are going to be in electrical equilibrium with one another. Okay, so all of these points that I'm colouring in orange, these are all the points of the cytoplasm. They're all going to be in electrical equilibrium with one another, and what that means is they're all going to have the same electrical potential. Okay, so if this little man comes into the cytoplasm of the cell with this machine and wanders around the cytoplasm of the cell, he can go and stand wherever he likes within the cell, and he will get the same number on his screen. Okay, so the electrical potential is the same in all of the little crevices of this cell, basically. Okay, so what I will do is I'll call the electrical potential within the cytoplasm capital E for electrical potential, and I'll subscript it I for intracellularly. Okay, so this stands for electrical potential intracellularly. Now, all of the points of the extracellular fluid, which I'm going to now highlight in red here, these will also be in electrical equilibrium with one another. Okay, so if the little man wanders around the extracellular fluid, the number on his screen will not change because all of these points have the same electrical potential. And the electrical potential of all of these points in the extracellular fluid collectively we'll call capital E for electrical potential, and then we'll subscript it E for extracellularly. Okay, right. Now here's the biggie. These two numbers are not necessarily the same thing. In fact, usually they're not. Okay? Usually there is an electrical potential difference across the cell membrane. Okay, now what does that mean then? So, the concept of an electrical potential difference, strictly speaking, should have a direction attached to it. Okay, so Electrical potential difference is also called voltage, and strictly speaking, when people talk about the electrical potential difference or the voltage across the cell membrane, what they strictly speaking should say is the electrical potential difference or the voltage from extracellular to intracellular. Okay, so strictly people should give a direction in which they're going. However, it's usually dropped. Usually people will just talk about the electrical potential difference or the voltage across the cell membrane, and they'll drop the from extracellular to intracellular, because it is assumed that everyone knows that they mean from extracellular to intracellular. Okay, so what then does this whole thing actually mean then? It means if this little man was sitting in the extracellular fluid, okay, so the little man starts off in the extracellular fluid, and he will have some number on his screen, okay, so whilst he's sitting there, he'll have some number on his screen. This will be the electrical potential extracellularly. It will be this number. Now, if he moves then from extracellular to intracellular, this number will change, okay? It will go from showing him the electrical potential extracellularly to showing him the electrical potential intracellularly. Okay? What the electrical potential difference from extracellular to intracellular means is how much would this number change on his screen if he moved from extracellular to intracellular. Okay, so let's now write the electrical potential difference from extracellular to intracellular in terms of the electrical potentials extracellularly and the electrical potentials intracellularly. Okay, so this is going to be the new electrical potential then, which is the electrical potential after he's moved, which is the electrical potential intracellularly. Okay, so once the little man has moved from extracellular to intracellular, the number on his machine afterwards will be the electrical potential intracellularly. Okay, minus the electrical potential extracellularly, which is the number on his screen before he moved. Okay, that tells you how much the number on his screen changes when he moves from extracellular to intracellular, which is what is meant by the electrical potential difference from extracellular to intracellular. Okay, so this formula here gives us the electrical potential difference across the cell membrane in terms of the electrical potential intracellularly and the electrical potential extracellularly. Now, usually this number is kept at negative 65 millivolts. Now, before we go any further, let's discuss the fact that the electrical potential difference across the cell membrane is going to have the same units as electrical potential. Okay, so electrical potential has the units of volts, okay, and we are just taking one electrical potential and subtracting off another electrical potential, and therefore this difference that we've now got is also going to be measured in volts. Now, millivolt just means a thousandth of a volt, so if you like, you can replace this with negative 0.065 volts, and it means the same thing. Okay, right. So what this means is that if the little man was to move from extracellular to intracellular, the electrical potential that is shown on the screen of his machine would change, and it would change by negative 0.065 volts or negative 65 millivolts. So the number on his screen would go down, and it would go down by 65 millivolts. This number here, the electrical potential intracellularly, is lower than the electrical potential extracellularly, and it's lower by 65 millivolts, usually. Okay, so this is known as the resting electrical potential difference across the cell membrane. Okay, now let's discuss our Ampa receptors. So let's draw another picture up here. So here is our Ampa receptor that is on our postsynaptic cell. Okay, so our postsynaptic cell membrane here currently has this resting electrical potential difference across it, okay, of negative 65 millivolts. So the electrical potential intracellularly is 65 millivolts lower than the electrical potential extracellularly. When the presynaptic neuron releases glutamate onto the postsynaptic neuron, what's going to happen is the glutamate is going to act on these Ampa receptors, cause them to open, and this is going to cause a depolarization of the electrical potential difference across the cell membrane. And I want to explain to you what that means and how it comes about. Okay, so here is our Ampa receptor here, which remember is a tetramer, either one of those homotetramers, one of those three homotetramers that we've discussed, a GluA1 homotetramer, GluA2 homotetramer, or a GluA4 homotetramer, or it's one of these diheterotetramers that's written up here, where you take some GluA2 subunits and then some of one of the other three types of Ampa receptor subunits. Okay, right. So basically what's going to happen is glutamate is going to be coming from the postsynaptic cell, from the presynaptic cell, okay, and the glutamate is going to bind to the 4 ligand binding domains of our Ampa receptor here. So remember, our Ampa receptor is this tetramer and therefore has 4 separate subunits. Each of these subunits has its own ligand binding domain, which means that you're going to need 4 glutamate molecules, one for each one of the ligand binding domains of each one of the 4 subunits, in order to activate your Ampa glutamate receptor. Okay, so when 4 glutamate molecules come and bind, so one to each of the ligand binding domains of each of your 4 subunits here, that then will cause the Ampa receptor to open. Okay, now when the Ampa receptor opens, it is permeable to sodium, potassium, and usually, well, sometimes calcium. Okay, so the calcium permeability depends on whether it has GluA2 subunits within it or not. Okay, so if you don't have GluA2 subunits in you, you will be permeable to calcium ions. Now, there are only 2 Ampa receptors that we have seen that don't have GluA2 in, which are the GluA1 homotetramers and the GluA4 homotetramers. All of the others do have GluA2 in, because remember, the others were the GluA2 homotetramer, which definitely has GluA2 subunits in. All 4 of them are GluA2 subunits. And then all of these diheterotetramers, they all have GluA2 subunits in. So the only ones that we've seen that don't have GluA2 in are the GluA1 homotetramer and the GluA4 homotetramer. Okay, so those will be permeable to calcium, whereas the others aren't really permeable to calcium. If you've got GluA2, you are not permeable to calcium, or at least your calcium permeability is feeble. Okay, right. So we'll try and keep this general. So we'll discuss it for the sodium and potassium, but we'll also mention calcium. Okay, so we'll focus on sodium and potassium, and then we'll add calcium in at the end, if you like. Okay, right. So when this channel opens, then it's going to allow sodium ions to move through it, potassium ions to move through it, and also calcium ions to move through it. And we now need to discuss which direction are we going to get those sodium, potassium, and calcium ions moving. Okay, right, so we'll start off with sodium ions. Okay, so we need to know the concentration gradient across the cell membrane, and we also need to be aware of the electrical gradient, which at the moment is negative 65 millivolts. Okay, so the intracellular electrical potential is lower than the extracellular electrical potential by 65 millivolts. So what's the concentration gradient then? Well, the concentration of sodium extracellularly is around 145 millimolar, whilst the concentration of sodium intracellularly is around 12 millimolar. Okay, that means that there is a 12-fold gradient in sodium across the cell membrane. Okay, the concentration of sodium extracellularly is 12 times higher than intracellularly. That means a sodium ion is 12 times more likely to hit from the outside and go through inwards than a sodium ion is to hit from the inside and go outwards. Okay, so this is favouring a net movement of sodium into the cell. In addition, you have to bear in mind the electrical potential difference across the cell membrane, because sodium ions are positively charged particles, so they're going to pay attention to the electrical potential difference that you have across the cell membrane. Okay, positively charged particles want to be where the electrical potential is lower. Now, currently, the electrical potential is lower in the cytoplasm rather than in the extracellular fluid. It's lower by a whole 65 millivolts. Okay, so actually, In this case, the electrical gradient is completely in agreement with the concentration gradient. The concentration gradient says go in, the electrical gradient also says go in. So basically they'll work together to produce a nice movement of sodium into the cell. So the net movement is going to be sodium is going to come into the cell. Okay, so that's going to happen for all Ampa receptors, whether they contain GluA2 or not. When you bind those four glutamate molecules to the four subunits of our Ampa receptor, and that causes the Ampa receptor to open, you're going to get a movement of sodium into the cell. Okay, let's now do potassium. The potassium concentration extracellularly is around 4 millimolar. The potassium concentration intracellularly is around 155 millimolar. Okay, so that means that you've got around a 40-fold gradient of potassium favoring the movement of potassium out of the cell. Okay, so the concentration gradient says go out. However, remember the electrical gradient. Potassium ions are also a positively charged ion, so they want to go where the electrical potential is lower, which is currently the intracellular compartment. So they want to go into the cell if they're just obeying the electrical gradient. Okay, so this one's a bit more of a conundrum. Which wins? Does the concentration gradient that says go out win, or does the electrical gradient that says come in win? Well, basically it's the concentration gradient. Okay, the concentration gradient of 40-fold is stronger than the electrical potential difference of negative 65 millivolts across the cell membrane. Okay, in actual fact, if you wanted to completely stop the movement of potassium out of the cell, okay, because of the concentration gradient, you would need to take the electrical potential difference across the cell membrane down to negative 85 millivolts. Okay, so if the electrical potential difference across the cell membrane was negative 85 millivolts, i.e. the intracellular electrical potential was 85 millivolts lower than the extracellular electrical potential, then it will be strong enough to completely stop any net movement of potassium out, even with this same concentration gradient that we've got here. We are only at negative 65 millivolts, so this is not strong enough to completely stop the movement of potassium out. However, it will weaken it considerably. Okay, so you're not going to get that many potassium ions going out, so I draw a little arrow there. Now, just for an understanding point of view, if we took it below negative 85 millivolts, if we took it down to, let's say, negative 95 millivolts, then potassium would actually start moving in, basically. Okay, so this is called the reversal potential, or the Nernst potential for potassium across the cell membrane. It's the electrical potential difference across the cell membrane at which there will be no net movement of potassium across the cell membrane, even with this concentration gradient. Okay, right, and it's often abbreviated to V for voltage and then K for potassium, and usually people put a positive to remind you that potassium has a positive charge. Okay, right, so basically if we just start off with the Ampa receptors that don't conduct calcium, what's going to happen? Okay, well, you've got a bigger amount of sodium coming in than potassium going out, which means that overall the net movement is positive charge into the cell, because both the sodium and the potassium ions carry a positive charge. Okay, so if we're moving this many sodium in and this many potassium out, then we're going to be moving a net amount of positive charge in. If you take the orange arrow and subtract off the blue arrow, you'll get the net amount of positive charge that is coming in. Okay, now this positive charge is going to affect the electrical potential difference across the cell membrane. Okay, now before we say how this is going to affect the electrical potential difference across the cell membrane, we might as well add in calcium for those that do have calcium, because it's not going to change this result that the net movement is inwards, it's just going to make it bigger, basically. Okay, so the concentration gradient then of calcium. Calcium's concentration extracellularly is around 1.5 millimolar, and calcium concentration intracellularly is around 100 nanomolar. Okay, that's a 15,000-fold gradient of calcium across the cell membrane. Okay, so a calcium ion is 15,000 times more likely to hit this ion channel from the outside and go in than it is to hit from the inside and go out. In addition, calcium is a positively charged ion. It wants to go where the electrical potential is lower, which is the intracellular compartment. So in addition, the electrical gradient actually agrees with the concentration gradient, so absolutely you're going to get a movement of calcium into the cell. Now you might think, well, this is going to be a gargantuan movement of calcium in a 15,000-fold gradient, but you do need to bear in mind that the concentration of calcium is quite low extracellularly when you compare it to the concentration of sodium, okay? So the movement of calcium isn't going to dwarf the movement of sodium, okay? But it is a considerable thing to add in there. Okay, so you're going to bring in your calcium like so. Now this, all the effect that this will have is that you'll have a bigger net movement of positive charge in, because you've added these calcium ions that are flowing in, and they're bringing two positive charges with them. Okay, so the net result is still that you've got this positively charged current coming into the cell. Now this has a fancy name. Okay, this is known as the excitatory postsynaptic current. Okay, or it's sometimes just called a depolarizing current. But to be fancy, we can use the excitatory postsynaptic current. Now, do not be afraid of this name. Firstly, let's discuss what a current is. A current just means a movement of charged particles. Okay, so whenever you've got a movement of charged particles across the cell membrane, that's called a current. Okay, this is certainly a movement of charged particles across the cell membrane. Postsynaptic because it's occurring on the postsynaptic cell, excitatory because it's going to make it more likely that this postsynaptic neuron is going to fire an action potential. And they're usually abbreviated to EPSCs. However, to be honest, I think a more useful name for this current is to call it a depolarizing current. Okay, because that's the most direct thing that this current is actually going to do. It's going to depolarize the electrical potential difference across the cell membrane. Now let's discuss what that means. So, if we are bringing positive charge into the intracellular fluid, okay, so we're bringing positive charge into the intracellular compartment, what effect is that going to have on the electrical potential within the intracellular compartment? Well, basically, when you dump positive charge in an area, that raises the electrical potential of that area. So bringing in positive charge and dumping it into the cytoplasm is going to raise the electrical potential of the cytoplasm. So this number is going to go up, basically. Now, we didn't just magic this positive charge out of nowhere. We got it from somewhere. We took it from the extracellular compartment. So actually what we're doing is we're picking up positive charge from the extracellular compartment and we're dumping it into the intracellular compartment. Now when you dump the positive charge into the intracellular compartment, we've just discussed that that will raise the electrical potential intracellularly. But when you nab it from the extracellular compartment, that will lower the electrical potential extracellularly, because when the positive charge was there, it was boosting the electrical potential. So if you're taking it away from there, obviously the electrical potential is going to go down. So this depolarizing current that we've got, which is this movement of positively charged particles from the extracellular fluid into the intracellular fluid, this is going to reduce the electrical potential extracellularly and raise the electrical potential intracellularly. Now, what effect is this going to have on the electrical potential difference across the cell membrane? Well, let's firstly do it intuitively, and then we'll go to the formula and check that the two add up. So intuitively, the electrical potential difference across the cell membrane started off at negative 65 millivolts, which meant, remember, that if you moved from extracellular to intracellular, you suffered a step down in your electrical potential. You stepped down from an electrical potential of this extracellular electrical potential to this electrical potential intracellularly, and I should stress we don't know what these absolute values are. All we know is that the step down was by 65 millivolts. However, now we're lowering this intracellular electrical potential and raising this we're lowering the extracellular electrical potential and raising the intracellular electrical potential. So we're bringing this one down and this one up. So to draw a picture of this, if this is the electrical potential extracellularly, and this is the electrical potential intracellularly, previously we've been stepping down by negative 65 millivolts. Okay, but now what we're doing is bringing this number down here and this number up. So the amount that we're going to step down is going to get smaller. So the electrical potential difference across the cell membrane is going to become less negative. Okay, so the step down is going to get lower. Now, the fancy word for the electrical potential difference across the cell membrane becoming less negative is to say that the electrical potential difference across the cell membrane has been depolarized, or the membrane has been depolarized. And we would call the action of depolarizing the electrical potential difference across the cell membrane depolarization. Okay, right. So, seeing this from the maths then, we have increase the intracellular electrical potential. So this number's gone up because we're dumping positive charge into the intracellular compartment. We decrease the electrical potential extracellularly. So this number's going up. The number we're subtracting off is going down. So of course this number's going to become less negative. Okay, so the overall effect then of the Ampa receptors is that when glutamate binds to the Ampa receptors, what that triggers is it triggers them to open. They're going to allow this depolarizing current, or this excitatory postsynaptic current, through them, which will depolarize the electrical potential difference across the cell membrane. And this depolarization in the electrical potential difference across the cell membrane also has a name. It's known as an excitatory postsynaptic. So EPS again, just for this excitatory postsynaptic, and then P for excitatory postsynaptic potential. Okay, so that's the name for the depolarization in the electrical potential difference that can be measured. So it's the name for the change in the electrical potential difference that we see. Okay, so I think we'll call it there for this video. My voice box needs a rest. And in the next video, what we'll do is we'll discuss the reason the Ampa receptors are called the Ampa receptors, which is this famous agonist called Ampa. Okay, and we'll discuss the structure of that and the horrendous full name for it. And then we'll move on to the kainate receptors, which will take us less time because the kainate receptors have a very similar function to the Ampa receptors. We'll then say a few words about the delta receptors, and then we'll move on to the Nmda receptors.